Or not? Okay. Is that good?
Yep.
Okay. We starting?
Yeah, we're ready.
Okay.
Yep, you're live.
Hi, everybody. Sorry for the delay. I'm Dr. Allyson J, medical oncologist at Weill Cornell Medical Center, and I'm here today with Dr. David Tuveson of the Cold Spring Harbor Laboratory, and we're here for our very first Facebook Live event on the importance of research in pancreatic cancer. Thank you all for coming, and thank you for your patience, and we're going to get started. I'm going to turn it over to Dr. Tuveson.
Well, good evening, everybody. Thanks for tuning in for a discussion about pancreatic cancer and how the research behind pancreatic cancer is the best way we have to change the plight of patients with this disease. Let's Win, as you'll hear from Dr. Ocean, is an organization that was started to inform patients about this disease and to show them that there is reason to have hope, that new treatments are on the horizon. I'm a scientist at Cold Spring Harbor, where I run a large laboratory. And my laboratory is supported by the Lustgarten Foundation, which is the parent organization which helped found Let's Win 1 year ago today. The purpose of the Lustgarten Foundation is to use research to change the outcome of patients by identifying better therapies for patients and by developing early detection methods so that the disease can be caught in an early surgically resectable stage. So here at the laboratory, my team of talented, enthusiastic young scientists have ideas that they test every day about how to accomplish those two objectives. We can't do this by ourselves. We're lucky to work with medical doctors like Dr. Ocean at Weill Cornell Medical College, where she takes care of patients every day. And so we have a joint partnership between science and medicine in efforts to improve everything for all patients. We've been at this for a year now in terms of Let's Win, and this is again, an organization that's able to provide terrific information to patients and their families. And we're hoping that this event today will serve in some ways to increase the awareness about the exciting research that's happening in the laboratory as well as in the clinic, and that patients can participate in some of this exciting research. So I'm going to turn it back to Dr. Ocean, who has completed a busy day of taking care of patients, so that she can give you her introduction.
So I am a medical oncologist, and I am also working with scientists to improve treatments that we are able to give to patients with pancreatic cancer. I run a lot of clinical trials, so we take the science and the research that is going on in the lab and translate that into potential treatments that can help patients in the clinic. And that area is called translational medicine, or translating the research. And so I work heavily with many scientists who are studying pancreatic cancer, and we all collaborate together to bring the latest science breakthroughs to the patients in a faster way. And really the bottom line of all of this means that the more patients that enroll in clinical trials, the more, the faster this science will get validated and also get into the hands of the patients to give them better treatment. So our focus is to publicize this research that's going on, to make sure that patients know about the clinical trials that are ongoing in pancreatic cancer, and then also to inform the patients of the ongoing clinical trials so that they can enroll in these trials and get therapies that are aimed at fighting the pancreatic cancer. There's different phases of research, and maybe I'll start by getting into the different phases of research that exist. What Dr. Tuveson does is in the laboratory, so that's called bench research or preclinical research, and that is research that is going on at the cellular level and also going on in certain model systems such as animal models or creating experiments on active live cancer cells in the laboratory. And I'm going to let him go into more detail about that. So that's the preclinical phase of research. And then once we have identified either a compound or a medicine or a technique that we want to test further and see if it helps patients, we have to enter it into clinical trials. And there are different phases of clinical trials. There's a phase 1 is the first phase, and that means that a clinical trial is being done for the first time in humans. And the goal of a phase 1 clinical trial is to discover that a certain treatment is safe for the patient. So we are not trying to test the different doses or even figure out what the right dose is. We're more trying to figure out what's the safest dose that we can give to patients, and is the dose of this compound or medicine safe? And so that's phase 1. Sometimes in phase 1 trials, we discover that an agent that we were testing for safety actually does look like it's working against the cancer, and we get hints of efficacy in these early phase trials. And then when that happens, that's when we take it on to phase 2. And phase 2 is when we're trying to find the correct dose of the compound to administer to patients, because we're seeing efficacy coming out in phase 1 and in the beginning of phase 2. Once we've established that this new drug has efficacy, then it's moved into stage 3 or phase 3 clinical trials, and that's when it's compared to a standard of care. And the standard of care are the already FDA-approved chemotherapy agents and sometimes targeted agents that are used in combination with chemotherapy. And then this new agent is tested again with the combination chemotherapies. And that is called a phase 3 trial. And if a phase 3 trial shows positive data, then that drug ultimately will hopefully get approved by the FDA for use in patients with pancreatic cancer across the board. Dr. Tuveson, do you want to add anything else to that?
Allyson, I think that was a terrific description of how clinical trials are available for patients today. As Dr. Ocean said, the critical problem is that many patients never participate in clinical trials who have cancer, particularly patients with pancreatic cancer. What we've been trying to do in the laboratory here at Cold Spring Harbor is to pick the medicines that we would suggest to Dr. Ocean to consider for early phase testing. And for this, as she mentioned, we've developed model systems of the disease. I'm going to use an analogy. When you're sick with an infection to the point where you go to the hospital, what the doctors do first is they try to figure out what type of infection you have. So if it's a pneumonia or a blood infection or a urinary tract infection, they grow that portion of your body in the laboratory, and then they ask, what is it that's grown and how do we kill it? That's how they choose the antibiotics that they would give to you. What we've been trying to develop here in the laboratory is if we could grow the cancer from a patient, in this case a pancreatic cancer patient, if we could grow it, could we grow it up fast enough and test enough different medicines so that we also could give information back to the treating doctors. And that would be Dr. Ocean. So that's an experimental approach that is basic research that we're performing right now in the laboratory. The models that we're growing of patients are called organoids because they resemble the organ of the pancreas, but they're not quite the same. But we get tissue and we grow it from a tumor biopsy, which can be taken by a surgeon who's doing an operation, or it can be taken by a radiologist or a gastroenterologist who uses a tiny needle to get a little sample. And so with these samples, we grow the tissue in such a fashion that we can study it quickly. And with that tissue, we test different therapies in such a fashion that it could be relevant to a patient. So we've been doing this now for several years, and we feel that we're at a point where the basic research can start to trickle into the clinic in the setting of a clinical trial, like Dr. Ocean mentioned. And we're in the process of designing such clinical trials right now, where we can test whether having this model of a patient, this organoid from their pancreas tumor, if that model would help pick a medicine so that when Dr. Ocean puts them on a trial, it's not a let's look and see trial, but rather we think it will work type of trial. We still have to perform the safety trials on new medicines, but many of the medicines that we'll be looking at are medicines that are already known to be safe because they're used for other medical conditions. And so the usual approach to clinical trials, I suspect, will be somewhat different, in this setting. But the bottom line is we want to use this basic science technique of having a model system called organoids to see if we can find medicines for patients that will help them right away in terms of stopping the disease and then causing it to shrink and eventually go away. That's, of course, the goal. But to do that, we need a strong partnership between science and the medicine. And the terrific thing about Let's Win is about providing hope to patients through providing information. And the information is the world has not ended. It's just that it's gotten tougher, and that we have to use all the information at our fingertips and link into all the people that can help give each of us the best shot at having quality time and at the same time quantity time. The goal of all of this is to turn pancreas cancer from a terrible problem into a treatable problem. And that's really the overall goal of all the work that we're doing right now in the laboratory, and encouraging physicians like Dr. Ocean to work with us to bring these ideas into the clinic. Dr. Ocean will tell you that, again, the serious shortage we have is that very few patients ever go on clinical trials. We do far more research in the laboratory that never sees, any action in the clinic. And a lot of it's because many patients just don't go on trials. And I think patients would go on trials if they had hope that the trial would help them. But as Dr. Ocean alluded to, we can't promise that the trial is going to help you, but we can promise that we're trying to help you by putting you on a trial. And we are looking for better ways to help you. We just can't predict if an experimental therapy will help a patient. And therefore, we have to go in unbiased and make sure that the medicines that we prescribe first aren't harmful. And then if they show that they can be tolerated and potentially can be helpful, then there are many ways of evaluating to make sure that we're not just being overly optimistic. And that's when you get to the higher level trials, the phase 3 and randomized trials that Allyson mentioned. But again, we're holding this session tonight because I think the science has gotten exciting, to the point where, practicing busy physicians like Dr. Ocean are thinking about incorporating some of the science in the way that she carries out studies. And this is, the moment that many patients with pancreatic cancer have waited for it. That it's not just next year, but rather we have some things right now that look very promising. And you shouldn't just settle for standard treatments as your only option. So the standard treatments we have available today are better than a few years ago, but they're still not good enough. And so we know we have to do much more to help our patients. So I think, Allyson, perhaps it would be good for them to hear what's it like for a patient when they come to see you at Weill Cornell Medical College, and how often is it that patients can qualify for trials when you meet them?
Sure. I mean, what you said was spot on. We have better treatments than we did years ago, but we clearly need to do better. And the reality is that this disease does not have cures. And we need, in addition to finding better treatments, we also need to find better early detection methods, which is another whole area of research that is ongoing. And we could talk about that in a bit. But when a patient comes to see me for a clinical trial, many times, unfortunately, they come to see me as a second or third opinion. And I almost wish that many of these patients would have sought out that opinion earlier on in their disease process, because I feel that they should be offered a clinical trial on day one of their diagnosis, because as you mentioned, the survival times are poor and we need to improve upon these therapies. And the only way we're going to do that is by enrolling patients in trials. So there are many patients I see, unfortunately, that come to me after many different opinions. I mean, I'm their third opinion or second opinion or whatever. And sometimes there's not a trial available at that point, but there would have been a trial available if they had sought out a trial earlier. So I feel that one of the barriers to getting on a trial is really knowing what trials are out there. A lot of people do not know what clinical trials are open and available to them. And Let's Win has actually paired up with PanCAN, the Pancreatic Cancer Action Network, to have their clinical trial finder on our website so that patients can actually go to the website and type in just a few details, and they'll get a list of clinical trials generated for them based on geography, where they live, based on there are other, you can search different variables about whether or not they're opening, they're about to open, etc. Whether or not certain treatments are allowed prior to going on the therapy. Some clinical trials want you to have no therapy first, and they want you to be, quote, untreated or treatment naive. And so all of that is listed on the clinical trials. And I really have to say that it is a well done site for looking at clinical trials, because if anyone has searched for clinical trials, and I even do this with my patients together as an oncologist, the whole process is very daunting and it's very overwhelming, and you don't know what trial is active right now, whether you're eligible for a certain trial, all these details that are really, really important that are hard to figure out when someone is first diagnosed. So if there's a way to streamline that process and to get patients or their caregivers or their friends or family who are interested in helping them out, seek out clinical trials for the patient that could potentially be good for them. Another bit of advice is if your own oncologist doesn't mention clinical trials to you, I think you have to, excuse me, speak forward and say, should I be thinking about a clinical trial? Is there a clinical trial going on in the United States somewhere that I could go on? And even if it's not being done in the center where that patient is, it's very well possible that it's being done in the next town over. There are many, many clinical trials that are ongoing. It's just that patients don't know about them. And so you just have to really get this information and ask your doctor about the available clinical trials. And if they tell you that there is not a clinical trial available right now, then I think that is a cue that maybe you should get another opinion even before you get treated to seek out a potential clinical trial. So I think that not knowing about the trials is a barrier to going on trials. Obviously, geography becomes the barrier, unfortunately, for some patients. It's hard for them to travel. They don't feel good. But I have done searches for patients who have told me if they're not in New York, they have relatives in Connecticut, or they have relatives in South Carolina, or they have relatives in Florida, and they could easily go down and get an opinion about a clinical trial somewhere else. So that is something to think about. It doesn't necessarily have to be at the doctor's office where you're getting treated. So I really, really want to hone in on that point, that you really should find out about what clinical trials are ongoing right when you get diagnosed. I think that answers the question of what I do with a patient who is newly diagnosed. Number one, I see if they're eligible for a trial. If they're not eligible for a trial, what I try to do is get the most information I can about the cancer, and what that means is doing genetics on the cancer, so doing some kind of genetic sequencing or genetic testing on the tumor itself. Many times this also involves doing blood testing the person's blood to see if they have an inherited reason to have pancreatic cancer. Is this something that runs in their family? But at a minimum, try to get the most information you can about the tumor, in addition to just being called cancer, meaning like what are the drivers of the cancer? What are the pathways that are activated in this cancer that potentially we have drugs against? Because if we can identify all of these actors or players that are driving the cancer, then we can potentially link the patient to a clinical trial that's testing the drug against that pathway. So it all starts with investigating the cancer for its, basically the genome of the cancer, figuring out what makes up the cancer so that we can identify better treatments for it. We can then match the treatments to the patient's tumor. And this is really the crux of personalized medicine, finding the right drug for the right patient at the right time. And all of this is being done through research. And I think that it's a pity that many patients don't get to go through this process of finding out more about their cancer, because I think we would have a lot more potential trials to offer patients if oncologists that treat this disease take a deeper look into the cancer itself.
So I think that was a terrific description of how patients would be assessed when they come to see you, Allyson. And I agree that the personalized medicine approach is the one that we as a medical community are embracing now. This notion that not every patient with a particular type of cancer is the same, but rather the molecular details of their tumor will help determine what are the best therapies that we could offer to them. And that's, in a way, the reason that the model systems are so useful. If you were to have a sample from a cancer and send it somewhere to be sequenced to look for the altered genes, the mutations in DNA, or the abnormal expression of RNA or protein, you would get back the list of potential pathways which may be relevant for your patient. That approach works very well when you have candidate pathways which themselves are terrific drug targets. In pancreatic cancer, the main driver of the disease is a mutation in a pathway called the RAS pathway, and the gene that's mutated is called KRAS, which is an odd term that was handed down by geneticists some 40 years ago when they identified the KRAS gene, which stands for Kirsten RAS. The mutation in this gene doesn't reveal a protein that you can drug with today's medicines. Rather, it reveals a mutant protein and pathway, which in most personalized medicine trials means you are going to be resistant to medicine. And therefore, what we tried hard to do with the pancreatic models here in the laboratory at Cold Spring Harbor is to identify therapies that are agnostic to the mutation that can kill the cancer cells. Similarly to how the microbiologists and the lab medicine folks at the hospitals identify antibiotics for bacteria. And we're hoping that this orthogonal type of information, which we call pharmacotyping, which is a jargon-esque word for drug testing. We're hoping that this orthogonal approach to the genetic testing that you mentioned will provide the most robust information to the physicians and the scientists taking care of patients. And I said it in that way because I think that's how the field of cancer medicine is going. The field of cancer medicine is not one where the oncologist is going to be able to figure it all out. It's getting much more sophisticated, and it's going to require a partner scientist to help take care of those patients. Just like is true for other disciplines, where it's not one. Think about surgery. Does the surgeon do everything? No. The surgeon can do nothing unless they have an anesthesiologist and other support staff with them to get the patient through the operation. Cancer is going to be the same way. We're going to need a scientist to be a partner with an oncologist. And the scientist may be a physician, but they don't have to be. Obviously, the pathologists and the surgeons and the subspecialty doctors will all be involved too. But the oncologist is going to go from the past where we had lists of drugs we would give patients with a certain type of cancer to a molecular oncologist where they know now not only do they have a type of cancer, but they have causes to their cancer that are going to be identified by gene sequencing, but also identified by pharmacological testing. And at least the way I think of the field, and I'm an oncologist too, but I spend the majority of my time as a cancer scientist. I feel that that's what's going to happen to our field, that it's going to take this sort of partnership between science and medicine to actually take care of patients. And, that's where we haven't gone before. Before in the past, it's as you described, the scientists had their laboratories somewhere quiet. They made discoveries, they published papers. And at some point or another, the things trickled up to the clinic and could be tested. I see a future where it's going to be different, where the scientists are going to be working with the cancer doctors, the oncologists. And I'm hoping that this is going to be a real win for patients. I believe that this organoid models that we've made is going to be maybe the first exemplar of how that can happen. We're still looking for that RAS therapy. As far as I know, nobody on this planet has one. There are some really clever people working hard on this, but it's a hot potato topic, meaning smart people that are well-funded, working in the private sector, work on it intermittently until the realization that in 6 months they're not going to have a drug that sets in, and usually a corporate decision is made, which leads to the cessation of the program. And so this is a hard problem. And RAS is not the only hard target we have in cancer. There are others too. But I think this is, again, where that marriage between science and medicine will be so critical. And I'm hoping that taking a very basic science place like Cold Spring Harbor Lab, which some of you know is one of the earliest independent research institutes, and working with bona fide, first-in-class medical centers like Weill Cornell, and having an intermediary like the Lustgarten Foundation and Let's Win will allow us to get to that next level of taking care of patients, which won't be slow and won't be quiet like it has been in the past. We need to take multiple shots on goal for individual patients, I think, to get us to that next level, not one shot. And as Dr. Ocean mentioned, patients a lot of times when they have cancer, they're too sick to get experimental trials, or they've previously been treated. Well, obviously we have to change that. We have to have new ways to assess patients for their ability to go on trials. And we have to have new ways of measuring whether, when they're on the trial, that the drug is working or failing. And this gets to the second big research area that I didn't mention before, but Allyson brought it up, so I should talk about it, which is the field of diagnostics. One of the arenas in diagnostics is early detection. So I mean, when I say diagnostics, I mean how you measure things. So you drive a car, but you're looking at all the gauges. You're trying to make sure you have enough gas. You're trying to make sure is the temperature too high. You're trying to make, now you got these computers in your car, so you're like looking to see where you should go because you don't know how to use maps anymore. But anyway, that's the same thing, with our patients. We need those things. We need the gauges, the fuel gauge. We need the oil gauge. We need the temperature gauge. We need the map to tell us where to go. We don't have any of that stuff. We're lucky. The things we have today are arcane, and they don't help us fast enough take care of our patients. And so one of the best things would be if we had a test that would tell us that a patient has a tumor when the patient doesn't know about it. So meaning a small tumor, say the size of a cherry, not that big, but big enough for someone to see on a scan or a blood test, that when you go for your annual physical, there's some method for finding it. Patient feels fine. If we could find the tumor when it was the size of a cherry, that might help patients, but it would really help them if we could find them when it was the size of a blueberry or a pea. That's what we want to find, that little pea that's our pancreas tumor. When we meet patients today, the tumor is the size of a golf ball, and it's too big. Surgeons say they can't remove it safely, and usually the cancer by that stage is spread through their body, and it's a difficult situation. And some of you listening to this, you'll say, that's me. And I understand that. And it would have been better if we could have met you when you had a tumor the size of a pea, but we don't have that technology today. And so that's one of the other things we work on in the basic science side. How do we see the pea that's the pancreas? How do we see that little thing? Well, some of you have had scans, you've had x-rays, you've had CAT scans, you've had other things, ultrasounds, MRI, whatnot. These are helpful for certain things. They usually won't find a tumor the size of a pea. But if it's the size of a cherry, they might see it. But I've told you that cherry's too big. We want to see the pea. And so one of the things that we've been doing here in the laboratory is figuring out ways to do that. And I run a lab here at Cold Spring Harbor, a very large lab, full of brilliant people. But we're not going to be first in class in everything. And so we try to partner with others on the outside who might have great ideas. And through the Lustgarten Foundation, one of the distinguished scientists that we support is named Bert Vogelstein. He's one of the grandfathers of cancer medicine and cancer oncology. He's at Johns Hopkins University. He's discovered cancer genes that some of you may have heard of, like P53 and APC. APC is the gene of colon cancer. And he's been working with us in the Lustgarten Foundation to develop an early detection test. And he's got a clever approach looking at blood to look for this earliest evidence of pancreas cancer. He's trying to get a test just like in the airport where they have those tests where they can find traces of bad things on your luggage. He's trying to develop that test for pancreas cancer using drops of blood. And early indications are that he's onto something. He's also working with some clever folks to do the challenge that I said is impossible. Use scans to find the P of pancreas cancer. So he's got very brilliant people that he's working with at Johns Hopkins to try to accomplish that. Here in my lab, we have different approaches. One day on another television moment, I can tell you about that. But the bottom line is, as Dr. Roshan was saying, if we had better ways to see the cancer early, we would help a lot more patients. And we're not here tonight to tell you we have the better ways, other than we're working like heck to try to find the better ways. And I do agree with her. If we could pick it up early, that would help a lot of folks right away. And so that's, an equal part of our efforts here in my lab and here through the Lustgarten Foundation generally. I think those type of approaches will help because it's a diagnostic to see it early. the other diagnostics I used metaphorically before, the fuel gauge, the oil gauge, the temperature gauge, etc., we need them too. And so we need scientists who aren't necessarily biological scientists or medical scientists, but rather physical scientists. So physicists, chemists, mathematicians, engineers working on this too. And they're clever folks. They like to find a new star in the sky. We want them to find the P in the pancreas. And some of them are picking up that challenge now. So Allyson, this was the other research topic, better therapies, better diagnostics. I think that's how we can help patients in the short term. I think part of our show is going to be that the patients had this chance to listen to us and throw questions at us. I'm not sure if we have any patients doing that, but I've just been passed a piece of paper.
We do have some questions that have come in. Some of them are a little specific, so I'm going to try to generalize them because I think they're just that everyone probably wants to know about them. One of the questions that came in is, are there any clinical trials for stable disease? Someone is stage 3, their cancer is inoperable due to various reasons for they can't have surgery, probably due to the involvement of the blood vessels, and they've had chemotherapy and they've had radiation, and now what? And they're stable. So it's an awesome question because that's an area of research that we really need to delve further into, because as I say to a lot of my patients who are stable on therapy, that stable is good, but we have to, in this time of stability, we have to figure out what's going to be the next step that we're going to do. And there are some clinical trials that look into newer treatments when patients have stable disease. But the person who asked this question is right. Most of the trials that are out there are for people that have progressive disease, so their disease is changing, and therefore it's to try to slow it down from it changing or growing rapidly. But as researchers that are making up these clinical trials, we really do have to write more clinical trials for patients that are stable and to figure out ways to keep them stable rather than just allowing patients who are progressing to go on the trials. Now, I can't specifically answer the question as to which trials are for stable disease, but stable is a very good time to try to find out more about the cancer. So if someone hasn't had genomics done on their tumor yet, that will be a time. They're stable, they're doing well, they're feeling good, hopefully. That will be a time to maybe go through that process of getting more information about the genomics of the cancer so that it can help us figure out what the next step would be, whether it's a trial, whether it's a change in therapy. So keep in mind that all of these procedures, biopsies, sequencing, etc., take a lot of time. And it's not just we give the idea to the patient, oh, we're going to sequence your tumor, and then we get the results the next day. It doesn't work like that. Many of my patients are very frustrated because unfortunately these tests take weeks to even a couple months to get back. And obviously time is what we all need, and time is not on our side when you have a disease that is incurable. I get that. But the more we do these tests, the more we are proactive about doing them earlier on in the disease course, the more we'll have this information available to us to guide therapy. And so that's really what I just want to say about doing this, searching for a trial for stable disease. If there isn't a trial for stable disease that exists out there, then your doctor should be working on sequencing the tumor to know what's going on with it for the next step. Another question has come in that says, what are the barriers to getting into clinical trials, and why aren't they being offered at more hospitals throughout the country? Well, I spoke a little bit about this already. The barriers exist in that they may be geographic. Sometimes the patient themselves doesn't feel physically strong enough to travel to go to a trial, so that can be a problem. I don't want to spend too much time on this, but basically there are resources for patients to help them get on trials, and one of these is the Clinical Trials Finder. Also, you could speak to any type of nurse navigator in your center. They're usually the ones that can help patients seek out clinical trials and get on clinical trials. So that's another resource that patients should tap into. Okay, another question has come in. This patient is asking, there's no evidence of disease on their scan, but the tumor marker seems to be rising, and they've been on chemo for a long time. This happens a lot, unfortunately. When a patient has been on chemotherapy for a while, the cells that usually were responding to the chemo or to the treatment that they were getting, the cells mutate and they undergo new genetic changes that then they stop responding to the therapy. And sometimes this is not seen on a scan just yet. It's seen in the markers. Sometimes the markers go up before we actually see it on the scan. So that's something to consider. Maybe it's a way of asking your doctor to get a scan sooner than normally would be done. Really, we have to make changes to therapy based on objective evidence of change in the tumor, and really we only get that from a CAT scan. So I think that a rising tumor marker would prompt an oncologist to get a scan earlier so that you could consider a new trial or a new treatment or a new line of therapy. So that's how I would best answer that question. Another question has come in about I'm just reading it to make sure I make sense of the question. After chemo radiation and the clinical trial using immunotherapy and radiation, the patient got the go-ahead to go have the Whipple surgery. So that means that they obviously had a response enough to attempt surgery. But unfortunately, at the time of surgery, the doctor had to abort the surgery because this is another thing that really is insidious about this disease, that the scan doesn't always match what's going on inside. And unfortunately, at the time of this patient's surgery, the doctor was not able to fully remove the cancer because it was found to be on the lining of the stomach, or what we call the peritoneum. And when that's found, we cannot remove the surgery, but the patient still does have options for treatment. Chemotherapy can be done. Clinical trials can be done. The same thing I told you about sequencing the cancer can still be done. Whenever you have surgery, you get a new biopsy, so then they can sequence that material to find out about that. And remember, we do have standard chemotherapy approaches available. So patients, there are regimens that are approved by the FDA that have clinical evidence of efficacy, and they should be used with the patient. And so I don't want anyone to get the message that just because they can't have surgery, they shouldn't have treatment. So I think that is the best way I can answer that question. There's another question that's out there, and maybe Dr. Tuveson can lend a hand in answering this one because it deals with a lot of the research that's ongoing right now. The question is, there's a lot of news out there about cancer immunotherapy for prevention and for treatment. Can we develop vaccines to prevent cancers that are not caused by viruses? I'm going to turn that over to Dr. Tuveson to answer that question.
Yeah, I mean, that's a prescient question, and it's one that the cancer community is attempting to address right now through a series of studies, first in preclinical animal models, but with the intention of testing it in humans. And so the notion behind this is that cancer is a disease where there are mutations in the DNA. One of the mutations that's relevant in pancreatic cancer is the KRAS mutation I mentioned previously. We know that certain types of cancer are viewed by the immune system as foreign compared to other types of cancer. These types of cancer are those diseases where occasionally you see spontaneous resolution of the cancer. And one example of that would be melanoma, where there were some patients in the past who would get an autoimmune disease whereby the pigmentation of their skin would go down. They would get a disease called vitiligo. And if they had melanoma at the same time, their melanoma would go away. And so the notion was that the immune system was attacking those cells that made pigment. And melanocytes are related to melanoma cells. And oftentimes melanoma is dark and pigmented like a mole. DNA sequencing showed that melanoma has over 1,000 mutations in the DNA, in genes. And you have 24,000 genes, 2% of the genome codes for the genes, 98% is the dark matter of the genome where all the regulation of gene expression occurs. So patients would have over 1,000 mutations in their melanoma genome, and you can treat those patients with immunotherapy. And when you do so, patients get better. Not all the time, but a lot of the time. And so former President Jimmy Carter, as some of you know, had melanoma recently that had spread to his brain, and he asked us to pray for him. And then he had the good sense to go to Emory Medical Center, where they treated him with immune checkpoint blockade antibodies. And I just saw a video yesterday of President Jimmy Carter talking about how we needed to do better with pancreas cancer, because he comes from a family where his brothers and sisters and parents died of pancreatic cancer. Well, he could only do that video because he was treated with the proper therapy for melanoma. And so all of us are asking, why can't we use that therapy in our cancer that we're fighting? Mm-hmm. In pancreas cancer, we have found through the work, again, of Bert Vogelstein and his colleagues at Johns Hopkins, Louis Diaz, and Young Lee, and Liz Yaffe, that we can treat a few percent of all pancreas cancer patients with the same approach. And these are the patients that carry a mutation in genes that are involved in regulating the stability of the genome. And if people have mutations in certain genes in the mismatch repair pathway, they get a lot of mutations in their whole genome because the genes that are supposed to fix the genome are missing. So you get lots and lots of mutations, and it's just like melanoma, thousands of mutations. These patients respond to the same immune checkpoint blockade when they have pancreas cancer. And this is great, a great result. And that's 1 or 2% of the patient. That leaves us with 98% of the patient to try to help today. And so one approach posed by this question from the audience is, what if we vaccinated before there was cancer? What if we knew KRAS was bad and we developed a KRAS vaccine? And maybe some other mutations that we know about in pancreas cancer. And that's a great idea. And that's an idea that people are pursuing. Mm-hmm. And we are going to be supporting through the Lustgarten Foundation somebody who's going to do something hopefully important in the early detection and cancer prevention space very shortly, working with Stand Up to Cancer. And we'll know about that by the early autumn. But you know, today the cancer vaccines we have are for human papillomavirus. And if you're in a European country, it's a required thing to go to school. If you're in America, it depends which state you live in because the states regulate that. And certain states don't approve it and others do. In any case, this is a vaccine your kids should get before they're teenagers because it prevents the bad type of the papillomavirus from taking resident in your child. Right. And this is the virus that causes cervical cancer in girls and head and neck cancer in girls and boys. And it can be totally prevented by a vaccine. But that's a virus that causes cancer. In pancreas cancer, we have no virus. We just have genes that cause the disease. And so vaccination is possible. It is a research question right now, I would say, not a clinical question yet. But developing immunotherapies that Dr. Ocean can have in her armamentarium is something we are actively pursuing, both on the scientific front as well as in clinical trials that she's mentioned. So I'm sorry I got a little long-winded there, but it is important that people hear that the advances in immunotherapy are not just going to be restricted to melanoma or certain types of lung cancer and certain types of kidney cancer. We imagine it's going to have beneficial effects on many more patients, but we have to still figure out the ways to get the immune system to recognize the pancreas cancer as being foreign. And that's a lot of the effort that's going on today.
I think that is a perfect time to start winding up because we have to be respectful of everybody's time. But I think we did get a lot of great points across in terms of educating patients about clinical trials and what we're doing in the lab to bring progress to these patients with pancreatic cancer. We are definitely going to have another Facebook Live event soon. And thank you for your patience in getting started. This was our very first event, and we had a little technical difficulty in the beginning, but we worked it out. And thank you to everybody who participated and to who asked us questions. And we are going to summarize all those questions, and we can post answers on our website so that you can see those questions and get the answers to them. That will be coming out soon. And if you have any further questions, you can write to info@letswinpc.org, and we will answer your questions regarding research in pancreatic cancer. And we're always trying to connect the patients with the best science that goes beyond standard of care. And so that's our goal every day. So please contact us with your questions, and we will do our best to get back to you. I want to say thank you so much to Dr. Tuveson for joining me today for this Facebook Live event on pancreatic cancer research. And Dave, I'll let you close out with a few words.
Okay. I also want to thank the audience for tuning in and for showing interest in this topic. Pancreatic cancer has gone from a quiet disease that people didn't like to talk about to one where everybody's networking and they are trying to learn anything they can, about this illness. Young people who have seen relatives or friends get sick with this disease are now being drawn into the field to perform research on pancreas cancer, because it's a Mount Everest that's never been climbed before. And so it's bringing in creative ideas from around the land, and that's helpful. For the patients of today, I hope that you realize that there is a lot of research being performed by laboratories in institutions across the country and overseas in efforts to find those new therapies and to find the early detection method and other technical breakthroughs. It will be a partnership with physicians that will allow us to test these ideas in the clinic. And the only way they'll be tested is if patients are enabled to participate in clinical trials. And we hope that the efforts that we have today will lead to these breakthroughs in the near term, so that we will have many more patients who are looking for that next therapy and that next therapy as we attempt to make the disease go away in patients. And I think through science and through patient community involvement, this is all possible. Just like it was possible to conquer TB and to conquer HIV. It's not different from those two diseases. But until those two diseases had their version of penicillin, it was tough. And so work with us while we look for our penicillin. And I think that you'll see things are going to be much brighter in the future.
Bye, everybody. Thank you.
Good night from New York.
Good night.